This is Dr. Dave Seftel reporting from ARVO 2014 for Macular News. And we're very pleased to have Professor Jean Bennett with us today. Welcome, Professor Bennett.
Thank you very much. I'm very pleased to be here.
Professor Bennett is going to be delivering the keynote address closing ARVO 2014 and introducing the world to some of the most remarkable research that has been reported that she has done and that others are working on that is transforming the landscape of gene therapy and stem cell therapy in macular degeneration and other fields. Would you tell us something about what you're going to be presenting?
Sure, Dave. First of all, thank you very much for inviting me, but I also want to mention that I'm just one person of a large team of investigators working on this very exciting work, and I will be telling what our team has been doing since 2005, when we first started planning to go to a phase 1 clinical trial, and what we're doing now, which is planning on completing what's called a phase 3 clinical trial, aiming to get approval from the FDA for this to be an approved drug that could be prescribed to anybody who could potentially benefit from it. So I'll be telling some of the behind-the-scenes stories, the experiences that we have had and that some of the subjects in our clinical trials have had, and how we have learned from these experiences and hope other people can learn from them so that they can move forward more quickly and safely towards developing treatments for other blinding diseases.
Now speak briefly about some of the tools that you'll be using and the tools that your colleagues will be using to take this research further.
Well, one of the big tools in retinal gene therapy and any gene therapy is the reagent that's used to deliver the DNA to cells. DNA is highly charged, and so it doesn't cross cell membranes easily. So you have to play a trick on it to package it in a shell, essentially, to get it across the cell membrane. And so the tool that we're using to bring DNA into the cells, the disease cells of the retina, is a recombinant virus called adeno-associated virus. This comes in multiple different flavors, and the wild-type version of this reagent has never been shown to cause disease in humans or animals. So this reagent delivers genes really efficiently and really stably to cells of the retina and does not cause a major inflammatory response. So we think that it has promise for delivering genes for multiple other conditions besides the one we're working on.
So if you were to name the top 3 things that have transformed this particular field of research, what would they be?
One of them is the progress that has been made over the past 20 years in identifying the genetic basis of different retinal diseases. There are now more than 250 different genes which have been identified, which when mutated can lead to a blinding condition. And in 1990, only one had been identified. So that's huge progress. Without that, we wouldn't know what gene to deliver. The second one is the knowledge of and ability to generate these recombinant vectors to deliver the recombinant DNA safely and specifically and stably to the cells in the eye. And the third would be surgical approaches with which to deliver the virus so that it specifically comes in contact with those disease cells and doesn't escape to other cells in the eye or the rest of the body.
In your particular laboratory, what are the things that you're working on going forward?
We're trying to extrapolate our results from this one clinical trial for a rare blinding condition towards other more common blinding conditions, most of which are inherited. And so we're harnessing the knowledge of how to deliver genes safely to cells to adapt to those other conditions. So in my laboratory, we have about half a dozen different disease targets in progress, targets that cause vision loss or blindness. And these are mainly genetic conditions. They're mainly rare, but they're actually prototypes for more common diseases. And we're hoping to bring those to clinical trial sometime within the next year or so.
Thank you, Professor Bennett. Is there anything else you'd like to add specifically for patients? What should they expect going forward in terms of the translation of your work and the work of others in the field?
That's a great question. I think the big message is there is a lot of hope out there. We are actually making, scientists and physicians are making tremendous progress now in terms of deciphering the causes of these diseases and then using rational approaches for treating them. And I would advise you to stay tuned to the progress in these conditions. There are many patient advocate groups out there who can give specific information about the status of particular conditions. For example, Foundation Fighting Blindness, Research to Prevent Blindness, the National Eye Institute. Many of these groups can tell you the state of the art. And if you have an inherited retinal degeneration, I suggest that you get genetic testing to try to figure out what the genetic basis of the disease is. Then you will be able to find out whether you have a condition that is about to be the target of a clinical trial and in which you might be able to participate.
Professor Bennett, tell us about the progress that's being made in terms of regulatory approval for human clinical trials, both in the United States and in Europe.
This is a really exciting time because the first human clinical trial using gene therapy was recently approved, and that was approved in Europe by the equivalent of the FDA called the EMA, or the European Medicines Agency. That is for a rare inherited condition called lipoprotein lipase deficiency. It's a metabolic condition, and there are strict rules in terms of how this particular gene therapy reagent can be used. Right now, there are no gene therapy drugs that are approved in the United States. But we are hoping that the results of our phase 3 clinical trial that's in progress right now for a rare form of a blinding disease called Leber's congenital amaurosis will lead to approval by the FDA of the first gene therapy in this country, a gene therapy specific for this particular condition, which is caused by mutations in a gene called RPE65. This would then be the first gene therapy approved in the United States and the first gene therapy approved for a retinal degeneration or a blinding condition in the whole world.
Thank you, Professor Bennett, one of the true pioneers in ophthalmology research. This is Dr. Dave Seftel reporting from ARVO 2014.